directly ask questions, and I encourage you to do so. But let's thank Cato for a fantastic presentation.
Thank you. Great. Thank you. Keep the water.
Okay, I'm good. Okay, so I'd like to now turn the podium over to our Kahn Professor of Chemistry, Professor of Material Science and Engineering, Professor of Biomedical Engineering, Professor of Pharmacology, Nathan Gianneschi, who will introduce the next speaker.
Thanks, Chad. It's a pleasure to be here this morning to introduce our second speaker of the morning, Jinwoo Cheon. Professor Cheon is H.D. Underwood Professor of Chemistry at Yonsei University, of course in Seoul, South Korea, where he's also the founding director of the Institute for Basic Science in the Center for Nanomedicine. Professor Cheon is known for his work in inorganic chemistry, in particular precision nanoparticle synthesis, particularly in the realm of magnetic nanomaterials for applications in therapeutics and diagnostics in cells and tissues. Professor Cheon did his undergraduate degree at Yonsei as well in chemistry. He did his PhD at the University of Illinois, Urbana-Champaign, and his postdoctoral research at UCLA and UC Berkeley. For his work, his seminal contributions, he's been awarded many accolades, including the Humboldt Research Award, the University of Illinois Sheth International Alumni Award for Exceptional Achievements, and the Samsung Ho-Am Prize, among many others. He's also a fellow of the Royal Society of Chemistry and the American Chemical Society, and as many of you know, he's the long-term editor at Accounts of Chemical Research. Please join me in welcoming Dr. Cheon.
So, is it working? All right. Okay, it's good. No. All right. So, thank you, Nathan, for kind introduction, and it is my honor and privilege to come Northwestern to give a IIN Symposium, and my thanks goes to the organizers and Chad. And my talk will be more into nanomaterials, but we have seen amazing success for the past century for using molecules and biochemicals to treat disease. And there are new interventions coming out based on physical stimuli such as light and electricity, and these are new, but it is quite effective for precision medicine based on spatial temporality and also device interfacing capabilities. So as you can see here, there have been very little study based on physical stimuli back in decades ago with only 7%, but for the past decade or so, it has grown to almost 30%. So you see huge advancement in this research field. And people have developed, for example, electrosuricals for therapeutics and light for and optogenetics for neuroscience. But one of the weakest points for these modalities or these technologies, very shallow penetration depths of their modalities. For example, the 90% decay depth is only a few millimeters, only a few millimeters. So now, in contrast, the magnetism can be quite advantageous with a big penetration depth with decay length 90% is 1,000 meters. So you just compare. The problem is that, unfortunately, there is no reliable no reliable intervention principles for biosystems. So you cannot use it. Now, this is just to bring it up. Which is you? I don't know. Which is you for typical weekends? We prefer to have some relaxed time, and tend to avoid strenuous workout. This is a mouse, naturally behaving, but it's changed to, work hard to run treadmill. Now, what would be the possibility to make this a sudden change? This is instantaneous, and this is a remotely promoted behavior. And actually, this was done by the magnetic promotion. And this is it will be the main theme of my talk today. We all know that MRI is a great invention for looking into the details of our brain. And brain is composed of neurons. And now, would you be able to modulate neural activities by using magnetism? If so, what would be the scientific principles behind this? People have observed that in some migratory animals can sense magnetism, in this case, quantum compass, to navigate their way to the destination. And there are something called magnetic receptors. You have to somehow sense magnetism, geomagnetic fields. And for prokaryotes, there are nanoparticles in magnetotactic bacteria. And for eukaryotes, there are two different models proposed. And one is a cryptochrome model where light in the retina is excited to generate by radicals. And the radicals somehow susceptible to the geomagnetic fields, and that brings into the neural activities. And the other model is based on magnetite iron oxide particles somehow linked into the cell membrane, and then depending on the orientation of the particles with the geomagnetic field, they may bring into the neural activities. But many of these results are not fully supported by scientists. It's very difficult to reproduce. And the magnetic receptors in those migratory animals is still elusive. What are those? What are the nature of those? It's still under investigation. Now, let me tell you about magnetic genetics. Magnetic genetics is a you could call it magnetic control of neural activities by magnetic receptors with genetic modifications. It was recently reported that when you have iron oxide in ferritin has a link to the cell membrane in 3,4 ion channel, and it could induce neural activities. But the subsequent results report that these are not reproducible. So it doesn't seem to work, and people look into the details. Why it doesn't work? And in fact, the iron oxide and ferritin core is made of ferrihydride, which is antiferromagnetic, meaning there are not many magnetic spins available. So the pulling force is very low, with below attonewton, and the potential energy is almost 10,000 times smaller than thermal energy, suggesting that there are not many magnetic spins to utilize. It's too weak to do anything. When it comes to the force scale, biology, it spans quite large area, large range from femtonewton to micronewton, and ion channel gating requires roughly 1 to 10 piconewton, which is very narrow force window, and it is highly likely that the previous study didn't provide enough energy to open the ion channels. So this is our magnetogenetic setup. It comes with a rotating magnetic field and magnetic receptor. This is artificial magnetic receptor. So we mimic this system with the operation of magnetic compass. Magnetic compass makes neither rotation to generate torque. This operation is truly long distance from South and North Pole, many 1,000 miles away, but it works. It's a long distance actuation, and our system comes with 3 components. One is magnetic field generator, the other one is a magnetic torque, and third is a piezo ion channel, which is mechanosensitive. The first is about magnetic torque, which is, we call it a component of artificial magnetic receptor. Octahedron shaped iron oxide nanoparticle of around 25 nanometer, assembled into a polysilicon bead of 200 nanometers through click chemistry, and we call this M-torque, magnetic torque. And it has a room temperature ferromagnetism with a weak magnetic coercivity, about 30 oersted, and it gives stable magnetic spins, which is very important. And now this assembled magnetic structure can have about 500,000 times stronger magnetic moment compared to ferritin. Now you need a magnetic field generator. And we devised a rotating magnetic field generator. It comes with multiple permanent magnets, as you can see there. There are a couple of them, and you arrange it into a circular array. And and then that will give you a uniform magnetic field. And typical magnetic tweezer has the field decrement is quite rapid with a power of force in distance. So the tweezer has very short working distance, less than 1 centimeter, a few millimeters typically. So, but once you have a multiple magnet array and precision design and the structure and so on, then you could enhance the magnetism with interference between those fields. So you could have a uniform magnetic field in a quite long distance. In this case, you could have about 1 meter of working distance, which is really important for any live animal biological studies. The third thing is about the ion channel. And now, because we are trying to utilize mechanical force to gate the ion channel, then you need a ion channel which should be really mechanosensitive. Now, the force in our body, in live animals, is very important from cell development to proprioception and body balance. And this is regarded as a sixth sense. This is very elusive, and this is intuitive, but somehow it's very difficult to sense. So therefore, the discovery of truly mechanosensitive ion channel had been really elusive. It wasn't until roughly 14 years the piezo was discovered. That's the first, the bona fide mechanosensitive ion channel. But the piezo in this case has 3 blades, and there is a channel pore in the middle. Now, the artificial magnetic receptors that we have developed as a piezo and M-torque. First, you need to genetically encode a neuron with a piezo by delivering the viral delivery of the gene. And once piezo is expressed, the labeling of a magnetic torque, the particle, is carried out by simple antigen-antibody interaction. You see the red dots are the expressed piezo-1 ion channels, and the M torques are on the right. The M torques on the cell surface, neuron cell surface. The M torque first align into magnetic field, rotating magnetic field. That's what you are seeing in the green dots are the torque, rotating torque, and the it generates torque, of course, and the force that you could generate ranges from a few piconewtons to tens of piconewtons in this case, and you could choose any force that you want to have. But piconewton is very weak, but it's very important for ion channel gating. I mentioned that you need 1 to 10 piconewton to gate the ion channel. So the piezo has 3 blades, and you attach M torque into the blade and apply lateral pulling of around 1.5 piconewton. That it opens the gate, and then the calcium influx is initiated. This is the setup, the magnetic generic setup you have. It comes with a rotating magnetic field. You can see that the outer rim rotation, and the calcium influx of the into the neuron is observed by X-RODA1 indicator, and the influx is quite robust and reproducible upon the magnetic pulsation. And the influx is tens of milliseconds, which is quite fast, and reversible. And this is a bioorthogonal process. So it only works with a purely mechanical sensitization. And once neuron is excited, it generates action potential and then brings into the brain activation. We tested our magnetic genetics into 4 different live animal studies. The cortex is the outer layer of brain, where the end terminal of neuron circuits, and it gives rise to simple behavior such as locomotion. And mid and deep brain have complex neural circuits for higher level behaviors such as feeding and parental behaviors. And then lastly, we tested our magnetic genetics for disease treatment, such as Parkinson's disease. So I'll tell you about this in detail. The first is about the locomotion of a mouse. And then we injected piezo and M-torque with a microseed into M2 region. M2 region is quite important in the execution of movement. So we selectively chose that area, and then here is the detailed experimental condition. And then we applied a magnetic field. Here you can see that the RMF, that's a rotating magnetic field. And then the mouse moves counterclockwise. And this kind of behavior has been well documented, actually. It's a known circuit process, because you activate M2 region in right hemisphere, that will prompt the movement of a left pole. And once you make an active movement of the left pole, you have to rotate counterclockwise. That's what you are seeing in this case. And you could do the opposite. Once you inject the mTOR and piezo into left hemisphere, it does counterwisely. So we show that, and this is bidirectional. It works quite nicely. Now, the second example is about deep brain and circuit level study. In this case, feeding. And the mouse is fasted, meaning that it's a hungry mouse. And it's ferociously. And now, when the rotating magnetic field is applied, it just loses appetite. And once you turn off the RMF, it comes back and it's again. So this is just one single mouse, and it's 10 minutes, 10 minutes, 10 minutes. It's just a sequential study. And we first injected piezo and mTOR into a region called the lateral hypothalamus and in deep brain. And in fact, there are at least 3 different neuronal types. You see that it's color-coded. And our behavior, controlled behavior of decreasing the feeding appetite is only possible with a selective activation of green neurons, which is glutamatergic neuron. So this is quite important because when you consider the brain has 100 billion neurons. And in addition to that, there are at least 150, more than 150 specific types of neurons. This is to make a very complex circuits and neural network processing. So this is barely known, and people are getting to know more about neural activities in terms of the circuit level. And for example, the glutamatic neuron is to go, the green light, and GABAergic neuron is to stop. So a typical area, you could see red and green at the same time, and should we go or should we stop in this case? There are 10 green lights here, 6 red lights. What would you do? You wanna hit the gas or you're gonna put the brake, right? So in the brain, I mean in nature, we already developed that we know how to select, and it's all about the specificity. And in this case, the neural specificity is quite important. Now, this is a third example. This is a deep brain multi-animal study. And here is a medial preoptic area. There are multiple different types of neurons. And in this case, we selectively activate the GABAergic neuron by injecting piezo1 and mTOR. And in fact, this mouse is a transgenic male. So this is a Cree mouse, they call it Cree mouse, and it only expresses piezo and GABAergic neurons. And then we tested to its parental behaviors. You see the arena comes with multiple mice, 2 adult mice, and 3 pups are behind the walls. The black dots on the right side, there are pups. Let me. So do you see here? And these are the pups, and these 2 are the adult mouse. And then this is a rather complex paradigm for neural circuit study. And this is about social interactions with multiple animals. I showed you just one mouse feeding, for example. It's one you and your food, and that's one-to-one interaction. But now we are talking about multiple subjects among live animals. And this is a large area, relatively large. It's about 1 meter size in natural environment. with barriers, and then steps, and walls, and so on. So this is more like a real situation. And then actually, this is a quite difficult paradigm for typical optogenetics. So the magnetic field is off at this moment, and the mouse, the green one has an artificial magnetic receptor, and the red is the control. And the field is on. You see that the rotating magnetic field. And then look at the green one. And then this area is the nesting zone in the middle. So the green one, actually showed promoted parental behavior to pick up all three pups successfully. And red one, didn't do anything. Pretty much, there's no motivation to go and pick up, because these are the virgin females. These mice had no interest whatsoever in pups. So that's why they were just ignorant at the beginning of this magnetic stimulation. This is about the treatment of Parkinson's disease with magnetic genetics. You all know that Parkinson's disease is a quite common neurodegenerative disease, and it's quite strong symptoms with motor dysfunction. Now, people have utilized electrical deep brain stimulation with the wire, simulating area called subthalamic nucleus, STN. It works, and this is one of the most well-known medical intervention. But it comes also with some limitations, such as it requires invasive surgery, the risk of inflammation due to the scar generation by the wire. And in fact, there is no cure of Parkinson's disease. It's just a temporary symptom relief. And then we tested deep brain stimulation with magnetic genetics by introducing piezo and mTOR into subthalamic nucleus area. And the mouse, Parkinsonian mouse, went through magnetic genetic stimulation for 30 minutes per day for 5 days. And and then the MG treated DBS in the red bracket, it maintains the balance through a rotor test, where the other mice couldn't keep up with the test, and also in the locomotion test, it gained almost 100% recovery in open field test. Then we interestingly observed a long-lasting therapeutic effects with a sustained recovery of around 35%, meaning that even though we stop MG stimulation, it still sustained, maintained the recovery of about almost 35 to almost 40%, and this could be due to the neuroplasticity and rerouting of damages, meaning that maybe it is fixing, it's curing into the disease, but the exact mechanism is still under investigation. So this is just a comparison of neuromodulation modalities. And EDBS, electrical deep brain stimulation, has a uncommon simulation, and it comes with a spatial targeting. And in fact, there is no need of genetic modification. Optogenetics is regarded as one of the best tools for neuroscience with a molecular specificity. Now, in contrast, our magnetic genetics, it has clear advantage of fully wireless and long-distance stimulation with molecular specificity. I demonstrate or that nature inspired artificial magnetic reception is operative. I hope I was able to convince you. And magnetic genetics comes with a spatial temporal wireless modulation with a therapeutic potential. And the magnetic genetics is not limited to neuroscience, and it could be useful for precision and intervention for any biological targets in deep tissue, because it has such a big penetration depth and non-invasiveness. So with that, I'd like thank IBS for the funding and my talented students and postdocs and my amazing collaborators. And thank you so much for your kind attention.
Thank you for an amazing talk. Let's take questions. The microphones will come around. Just stand up when you receive them.
Thank you very much for insightful talk. It's natural, as you mentioned in the beginning, that electric field is being used usually, and then comes the application of the magnetic field, historically in many situations like that. And we call ourselves, I'm an electrical engineer by training,
Okay.
and if I tell people that I'm a magnetic engineer, people may not know what I refer to. Your research makes use of the magnetic field. But you use piezo material, which
Right.
many of them are ferroelectric. So one would think
No.
if you make...
Okay, the piezo, actually, that's just name. It's a purely mechanical ion channel. So it was discovered by the Adam Patapoutian at Scripps, and he just named it as a piezo, because piezo is meaning the pressure, the force. So there are multiple different types of neurons. Some are voltage gated. It's very susceptible to the electric voltage. Some are chemical. Some are pressure related. When this the piezo is meant to indicate the ion channel, which is only responsive to the force, mechanical force.
Along that vein, would you think trying ferromagnetic material would have some advantage, or did you explore ferromagnetic?
Ferromagnetic materials. In fact, the particle that we used is a ferromagnetic. It's nanoparticle of let's say a few hundred nanometers. But the core magnetic particle is about 20 some nanometer size. It comes with a very weak magnetic coercivity. To have a stable magnetic spins at room temperature, you need you think about the magnetic compass. It's a ferromagnetic, and then it has to maintain magnetic spins at room temperature to serve as a magnetic compass needle, right? And then you rotate the geomagnetic field. Whether you cannot rotate, I mean, the Earth is rotating, but you cannot really rotate the Earth, right? So that's why when you have a magnetic compass, the needle rotates, because the needle is trying to align into geomagnetic fields, right? And then that force, the torque, is something that we are going to use, we are using. So simply you have a magnetic compass in your hand, just make it, billion times smaller, And that's what we are having. It's a neural nano magnetic compass, and then that regenerates mechanical torque, and then the torque actually is delivered into mechanosensitive ion channel named, for example, there are a number of different mechanosensitive ion channels, not limited to piezo 1 and 2. That's a very specific, mechanosensitive, and there are others. So that's why the gating process only driven by mechanical force. It's like, when you have a door, you open the door, and it's almost the same. But our body has that, that process. So it comes to our body, it's so complicated, so that's what, it took because, your body balance, when you walk in the dark, and then you touching your nose and when you close your eyes, and those are quite difficult to understand. And still, and then it's related, associated with purely mechanical sensation in your body, in our body. So that's one of them is a piezo 1 and 2.
Okay, one more quick question. Yep.
Yes, thank you for a very stimulating presentation and talk, no puns intended. So I think that Maxwell's equation proves that you cannot focus magnetic field, but you can produce a node of empty field.
Right, yeah.
Your approach, which is very interesting, could not target a specific location.
Right, you cannot.
But could you imagine the opposite? If you would have a stimulation where there is no magnetic field, then you could focus it and localize.
Yeah, that's there is the instrument and MPI, right? That's MPI, right? And right, you could, the focusing of, because it's a field, right? There are interference between the fields, and that's pretty much it. So you could make there is no magnetic field where the rest are. So, but we found that that instrumentation, development of instrumentation is too complicated. So and then it's much easier to use magnetic nanomaterials, and then let them just work with the field. But the field has to be constant. In biology, it's better to have a designated space with a high resolution. In this case, in our case, the spatial resolution of the targeted area is only a few micrometers. Only a few micrometers, but you cannot do with MPI. It's not, and it comes with a heavy instrumentation too. So, but here we use multiple permanent magnet system, and then the system is very simple and easy to use. So right, you could devise the system, and also you could utilize less magnetic, the area, spatial area, with setting of electromagnets and so on. But I think this is simple and easier, and we consider that option, many times. But still, we have no idea how to proceed in that way. But this works quite nicely, so we chose to stick to this case.
Okay. Let's thank Professor Cheon again for a fantastic talk. So we're going to get back on time and stick with our start time after this break of 11:05. So go ahead and have a coffee, and we'll see you back at 11:05. Okay.
Good morning, everyone. We're going to resume. So if you can go back to your spots. I'm Cécile Chazot. I'm an assistant professor and the Julia Wortman Professor of Material Science and Engineering here at Northwestern. And today it's my great pleasure to introduce Ting Zhu, our next speaker. Ting Zhu is a distinguished professor at UC Berkeley. She holds an appointment in both material science and chemistry. She's also a faculty senior scientist for the materials division in Lawrence Berkeley National Lab and one of the members of the Kavli Energy Nanoscience Institute. Her academic journey started in China, where she earned a bachelor at Dalian University of Technology and a master's from the Chinese Academy of Sciences. She then moved to the US, where she got a master's and PhD at UMass Amherst and did her postdoc training at University of Pennsylvania and at NIST, the National Institute of Standards and Technology. Her research really bridges polymer sciences, protein science, synthetic biology, and colloidal science. And her innovative approach is really centered around the rational design of functional materials with application spanning from the life sciences, the environmental sciences, and energy. She's a fellow of the American Physical Society, the American Chemical Society, and on the board of directors for the Materials Research Society. Her research has been recognized by many awards, including the 3M Non-Tenure Faculty Award, the DuPont Young Professor Award, the Office of Naval Research Young Investigator Program, and the Li Ka-shing Women Research Award. She also received recognition from the Camille Dreyfus Scholar Teacher Award and the ACS Arthur Doolittle Award. In 2009, she was recognized among the Brilliant 10 by the Popular Science magazine. And her team has won the grand prize in the 2021 Create the Future Design Contest and was recognized in 2022 for the Breakthrough of the Year Award in Science and Technology from the German Falling Walls Foundation. With this, join me in welcoming our speakers, where she will tell us if macromolecules are friends or foes.
Can I borrow a laser pointer? All right, let's see if I can get this through. OK, that works great. All right, so good morning, everyone. And it's indeed a great pleasure and honor for me to be here. And I was specifically instructed that I need to make the talk entertaining and to deliver a message. So when I thought, what will be the title, and I say, what do I know first? I know polymers, and in some cases, they're called micromolecules. Some of you think about micromolecules, you say, those are DNAs, which IIN is very famous for, proteins, and maybe we can get mRNA in there as well. And of course, when we start thinking about life, start thinking about who we are, That's where we think, right? So you have a protein, you have a biomachinery, translate the sequence, translate the information, and just to be in sync with Nobel Prize, I put an AI-generated picture of DNA and protein there. So hope you will find it entertaining. But in parallel, we also have to, some of you may think about polymers as synthetic, okay? And in fact, starting from 1920, when it was the first proposed micromolecules, you can look at the history, and then to this, pantyhose sold out when the nylon was first invented, to the famous word of plastic in that movie. And plastic is everywhere in our world, and probably more than often, in the last 10 years, perhaps 5 years, plastic has been really, I cannot say demonized, but this picture come out a lot. Put it this way, my son is only 10 years old. He sees plastic as this bird in the Monterey aquarium. He always said, those are the plastic waste. That's what you do in, mom and dad, that's what, the thing you do. So the question we want to ask ourselves is, where should we go? We're at the crossing point for polymers. Is it possible for us to turn our friends and no foe? And what are the future we're going to leave for our next generations? So many years, this is where we are. Biopolymers and synthetic polymers are running in parallel, okay? But what happened is that synthetic polymer really goes up. The slope of the growth exceeded what nature can accommodate. The growth rate, what you have in the biomicromolecules through million years of evolution versus what synthetic polymer is doing, are completely decoupled. That's where we'll end up. If we keep in that trajectory, we'll no longer have a Greenland period. What I would say is that perhaps we should think about design future materials, including nanomaterials, in a different way. We shouldn't treat it as a parallel and socially well responsible as a scientist. And rather what we're supposed to do is, many of you learning driving, you're trying to get into a highway. In the highway, there are very fast racing cars. They're running with their own rules. When you get on there, you must follow the speed. You must follow what the cars are already doing. By doing so, you're not going to get accident. And in this case, we'll say age doesn't matter. Plastic is only 100 years old, and biology has been doing this for millions of years through evolution. Perhaps there are things we can learn from it. And in fact, that's not something foreign. Many of us are doing exactly that. Biomimicry and biomimicry material has gone through, throughout the time that we're trying to engineer material. In nanomaterial, it's particular true. You can literally not find a paper in nanoscience not saying that this is bio-inspired. And many of us use either synthetic analog or, in fact, biological component. So now let's take a look at to see what are differences between biomicromolecule and synthetic biomolecule. From the chemistry point of view, if you look at the biomolecule, we know there are 20 amino acids and there's the backbones, and the backbone is amide chemistry. And that chemistry gives a lot of unique features associated with protein. I want to keep that in there. Synthetic polymer, on the other end, if you look at polyolefins or polyolefin-based materials, that is petroleum-based. Many of them have a carbon-carbon backbone. Of course, you can diversify that, and you have a lot of synthetic capability. You can even put non-carbon molecules into the backbone. And then we'll start thinking about the conformation. This is really, timing, is perfect. You just see how the protein structure get awarded, last couple of days. And I would say when you have a protein structure, this structure is always under the constraint of the amide backbone. And polymers actually also have a structure. Many of us in polymer will say, we call it polymer conformation. If you have a semi-crystalline material, you can get atomic structure. But in this case, what I would say is that your conformation is really reflected in ensemble of your bond angle and torsion angle distribution. If you look at 3D random walk, the conformation is dictated by the distribution of your torsion angle, and keep that in mind. In a sense, I always say the Ramachandran plot actually is a special case of commonly we talk about the torsion angle distribution. And of course, when you start thinking about synthesis, this is where, through many of us off track. Basically, we say, when you start to diversify the monomer chemistry, you are having to have a sequence. And the sequence specificity always come out because that's the dogma for biology. And how you're going to get this monomeric sequence precision in your synthetic polymer has been a really big roadblock for us to realize that. And yet biology has biomachinery, have ribosomes. They all sound really good. But I have to always think about one question. Is the hen comes first or the egg comes first? Who made the S1, S2 subunit? Who made those proteins that make biomachinery possible? If you are willing to go over that barrier, perhaps you can begin to challenge if the monomeric sequence is indeed necessary. And of course, when you start thinking about diversities, in this case, biology certainly roots. We look at the sequence space, we look at the chemical diversity versus when we look at the diversity in synthetic polymer. All right, so I'm going to tell you the foundation hypothesis of my research. And you don't necessarily have to believe it, but this is what we learned through about 7 years of basic study trying to investigate polymer and peptide interactions. We really look at the energy landscape for the interactions as function of distance. We also look at various contributions in terms of block lengths, etc. So this is what we get. We're basically saying that proteins are evolutionarily selected, and they should not be looked at for function as one single role that we can define, but rather they tend to be multifunctional. And the second aspect is that protein sequence function relationship is essentially reflect the evolution result under the constraint of the amide backbone. Okay? As you change the backbone chemistry, that particular relationship should be reconsidered. And the third part, it can be controversial, but this is what we firmly believe. The primary and secondary sequence structure in the protein may not be the prerequisite for synthetic polymer to replicate protein's function. And the part where particular interest is that we must increase the chemical diversity that currently are not commonly seen in synthetic polymer. And how we should design is really look at a segmental interaction that's reflected in the protein tertiary structure. And the last part we really should think about is how we should retrack the evolution path instead of bottom-up approach for proteins and engineer protein at the ensemble level instead of individual chain. So with that, let's start talking about as you increase the diversity of your monomer, how that relates to the information. We all know bioinformation. So what we see is that you have, let's say you have 20 amino acids. You have different monomers. And you have a polymer has a degree of polymerization, DP site. And if you have every site to have every monomer possibly goes in there, N to the DP is your possible sequence space. Okay, think about that in the entropy phase, entropy aspect. It's really large. And if you look at a sequence-specific polymer, this is where you are. That means each individual site, the monomer, has to be specific. And you have one probability, one sequence in there. And if you are willing to do an entropy, that's where entropy equal to 0. This is too high, this is too low. And when you begin to look at the proteins, we'll say somewhere in the middle. If you look at the probability of each monomer at the site, and that will give you an ensemble, that's exactly what the protein sequence, protein ensembles are being evolved in. And we all know that if you have a homopolymer, homopeptide, you are maybe somewhere in certain sectors of sequence space, and they get evolved out. They get deselected throughout. And in fact, in many cases, the material we design happen to be the one that gets deselected evolutionary. So synthetic, they turn out to be a good timing. There's a new development in the reversible deactivation, activation polymerization, and this is where we go. And that's where if you get into polymer chemistry 101, you know that I cannot control monomeric sequence, but I can control the probability of the monomer at each individual site. And if I do that, what I'm going to get is that the reactivity ratio of that monomer coupled with a monomer concentration locally to the first approximation is going to determine the probability for monomer in that. And then using that, I can control how I'm going to fill up the site as I'm doing polymerization. You can easily do a Monte Carlo simulation. It's going to get there. And what you synthesize is that in that batch, you're going to have an ensemble. That means each individual chain are different at the monomeric level, but they share one thing. That is a probability of the monomer at each individual site. And mathematically, you want to go back to the sequence, the number of sequence you get, you're going to see that you happen to be in the range that I was referring to. And what does it mean? What does this probability at site means? And that was a big question, right? So I put different monomers, I can control the probability and narrow the sequence space. How that translate into the physical meaning? So let me use one single chain to tell you what does it mean. What that gives you is that you can cluster the monomers together, and that is going to form a segment. And we actually have done this years ago. This is, many of you study polymer physics. You're going to do equivalence. Okay, we don't have to know the torsion angle distribution at each individual monomer. We cluster them. And that gives you equivalence. All of a sudden, you can use equivalent Fourier joint chain model to look at your radial gyration of your polymer. If you use that same analogy, you can look at the proteins. Proteins can be broken down into segments. That's how we do peptide design. Early days when we do de novo peptide design, we're going to have a loops, alpha helix, and beta sheet. And subsequently, we assemble it into a tertiary structure. If you start to combine those two and then get into a random copolymer, this is what you get. You're going to have a cluster of the monomers. Their accumulated hydrophobicity can be either hydrophilic, hydrophobic, or amphiphilic in the middle. And that sets up things really well. So Monica Ora de la Cruz was a Miller professor in my group. Back then, we worked together. And we're basically saying that if you have a polymer chain, can break down into the segment, and the local characteristic of that segment is going to help the chain to fold when they insert it into the lipid bilayer. And this, in fact, it works really well. We put a key residue, which happened to be a proton conductor, be able to hydrogen bond with proton. And we're actually getting a pretty good proton transporter. And Monica did simulation for us, if you can find my mouse, which I probably not. Oh, here we go. OK. So in this case, this is a few interesting things. Here is showing the water network. The segment inserted in the lipid is very static. And the one outside in the water is very dynamic. And the one at the interface, those short segments, happen to be somewhere in the middle. Not only you can spatially position that key residue, but also what you see is that you can access this dynamic heterogeneity that is really in the essence of folded protein. Not a static structure, but also dynamic, but rather the dynamic heterogeneity. If you do that, this is really open up opportunity. Now I can translate protein sequence at the segmental level instead of individual. The question is how I'm going to do that. This is where we work with data scientists. When we first started, we are very primitive. We say, let's translate a sequence into a binary. This really follows Steve White. Steve White did this hydrophobicity scaling associated with 20 amino acid. We say from the glycine, based on glycine, we can get either hydrophilic or hydrophobic. We can look at the block length. As simple as it is, actually works really well. It was detailed in that paper 2018. We did get a little bit better, given where the data science goes. So now what we do is we look at the segment of proteins, and we're using sliding scale that biophysicists use all the time. We break down a protein sequence into a certain segments, and we look at the overall chemical characteristic as well as sequence information of those segments. And this is where we get. So here, what do you see? The dot, green dots corresponding to a transmembrane protein, and the red corresponding to a globule protein. And we have a 2D principal component analysis. In the x-axis corresponding to hydrophobicity of that segment, and the y-axis tell you the blockiness within that segment. So by looking at the two-dimensional, in this case, what do we see is that, first of all, there's a clear transmembrane protein and water-soluble protein, their hydrophobicity segment distribution is different. But what is also interesting is that if you look into the blockiness of the PC2, if it's really blocky, it will possess space here and there. But what do you see is most of protein sequences are very much scrambled. Protein, evolution-wise, doesn't like to have a distinctive hydrophilic or hydrophobic patches because they have to function as a part of the community instead of for their specific functionality. And if you want to look at the typical homopolymer, block copolymer, they will be far out of here. And this is part of the reason that we hypothesize a lot of synthetic polymer are not biocompatible. They are problematic ecologically because they happen to be in the sequence space that has been deselected through evolution. So obviously, I didn't tell you which segment is doing what. We'll look at the population. We'll look at the dynamic range of the system. And we are really looking at the system compatibility at the random encounter level. We'll now look at specific functionality, for example. So what would be the best way to see you capture what nature decided to select? And we decided to use protein folding during cell-free synthesis. As you fold, you don't have a structure. You're not stabilized specific structure because you simply don't know what's come out. And isn't that what nature is, did a good, beautiful job. So what we did, we take all the units, all the functional elements, all the components, and we take it out of the cytosol. We backfill it with random copolymer, hypothesize that random copolymer we designed can capture what the proteins are doing, what are the random encounter, what type of energy associated during those events. And of course, if it doesn't work, I won't talk to you about it. In this case, we focus on transmembrane protein. And what you see is that the protein fold better than anything we're aware of it. But that's sort of not, you work on it, you send a proposal, you get it, that's good. What is really surprising is here. We use the methionine, the radiolabeled methionine, to look at the protein production. And surprisingly, what you see here is that for the protein production, we increase about 100% or more. That's indicating that even though you design with a protein sequence information, the biomachinery somehow also like that particular environment. And this is where, we talk about things are co-evolve. You're not doing it at one molecule at a time. You are really look at the whole population, look at for the benefit of the ensemble. And there are certainly some designability. So what you have is its map works really well, and you can use that to individually program your polymer for cell-free synthesis of various transmembrane proteins. Like here, there's a protein channel, there's that peptide-based some channel. This is basically, we take the sequence or take the information from the vendors, we get in, we try them out, and so far has been doing great. That's a random. And we ask ourselves, can you also look at things at both ensemble as well as a specific chain, specific pair interactions? So here are three things. This is a very busy slide. I'm sorry for that. So first of all, what do we get is we're going to look at the proteins or peptide that go through liquid-liquid phase separation to generate membraneless organelles. And then from there, we say, OK, the polymer does that. No surprise, many people have seen it. But what is interesting for us are threefold. First, we look at the viscosity of those droplets, those condensates, and the viscosity is very much similar to what you see organelles inside of the cell. And that basically saying that whatever the composition, whatever the water hydration level in there, it really recapitulates what you see in biology. Okay? And the second thing we did is we can use that to partition where the protein goes and subsequently enhance the thermal stability of the protein. And with the same polymer, with similar polymer, we can also encapsulate and compartmentalize single-strand DNA. And then when we fuse the droplets together, DNA complex, but we can control the T-half of that complexation protein. And if you start to think about regulated gene expression, to some extent, it's a very analogous. Okay, this is all non-cell, very synthetic. So this is also when you have a diverse student who are really innovative or resourceful under the cold chain logistic issue have a problem. I have one student in biology, and he basically said, I can't get my serum on time because the ordering process takes really long. You guys told me you can stabilize proteins. How about you give me some so I can expand the shelf lifetime of my serum? And that's exactly what he did. So he put FBS, put some polymer in there, put FBS in there. No refrigeration, leave it on the countertop. And it works, for a month, there's no problem. He didn't see any aggregation. And he subsequently used that to grow cell. And the cell grows really well. And what we like is that 2023, last year, Genentech obviously had a team working on this, and they used random hydropolymer to stabilize their high concentration antibody. And what is particularly rewarding I don't get any money, I have nothing to do with it. But what is rewarding here is that they actually adopt exactly the same chemistry, the methacrylate monomers. They just, do a large-scale synthesis to really screen out the side chains. And if I may confess, some people ask me, how did you choose those 4 monomers on the side? it was by random. It was not something I designed to get in there. And we study each of them as a homopolymer, and that's how we come up with the formulation. And I would say the possibility is unlimited. So we can certainly, give the possibility you can stabilize proteins, under reasonable buffer condition. So can you do it outside of the biological environment? And this is where we take advantage. We can have an enzyme stabilize it and nanoscopic disperse it inside of the plastic. We can make them to compost, within a few weeks under the compost condition. And what is interesting for a scientist here is that the enzyme are nanoscopic dispersed. They are embedded after the plastic breakdown, so so-called microplastic. And then subsequently, they can get into up to about 98% polymer to monomer conversion. And they break down not into a short chain polymer, but rather than small molecules. And the small molecule can be substituted, used as nutrition for the microbes. And of course, this is a process really compatible with a different ingredient because you trap the enzymes, and then you provide this little microenvironment like what they have inside of the cell. So in this case, we mix it together with some conducting fillers, carbon black, silver flake, whatever you can find on the shelf. And this mechanical engineer student is very talented. He made the whole thing to have a compostable plastic coupled with compostable electrode. So the whole thing in the end can be completely recycled. You can get your metal fillers out, and then you can redo the process. And the mechanical property is also quite good. All right, so this is Nano Institute. I'm going to spend the last couple minutes to tell you what we learned for the biology, how they really teach us or expand our thinking process for a synthetic nanomaterial engineering. So many of us are here, and obviously we want to hear. And I can tell you there is a gap, and the gap is here. I will explain to you in a minute. Okay? So synthesis-wise, you can pretty much synthesize just about everything here. Okay? Whether you can synthesize at a scale is still remain questionable, but we're slowly but surely getting there. This is where the challenge comes in. You nanomaterial design based on this triangle, golden triangle, chemistry function structure relationship may not be what you need for the device. And here is a barrier materials. And let's see if I wanted to make a barrier material for my organic photovoltaic to prevent oxygen diffusion or prevent water. In that case, you have to worry about interfacial roughness. Okay? So if I wanted to have a barrier material to prevent the VOC, I coat the walls so that you have the VOC emission. And then on top of that, if you wanted to look at the small molecule permeation within a barrier material, you have to think about the specific thickness, specific chemistry, and your defect concentration as well as defect type. Those basically expand your golden triangle with all the additional requirement. And we actually don't know where the facets will end up. Okay? But the key question is that from the science to technology, perhaps you should rethink many other factors. And this is where we think biology does really well. This is where the diversity comes in. This is where precision is embedded and has to be utilized in the realm of complete randomness. Besides enthalpic, intermolecular interaction, you really start thinking about entropy. Biology doesn't rely on one or two molecules. It relies on many. And if you look into any biological systems, it's never as clean as in your test tube. And there's a reason for that. Okay? For example, look at the cytosols. I don't even know what the content you have. I checked the blood composition. The list just goes on and on and on. There's so many of trace content in there. We have absolutely no clue what they are there for. But thermodynamically, as you increase the number of the ingredient and start to look at that, entropy comes into play. And mixing entropy becomes more and more important. Okay? And you see this in high entropy alloys. People put all the atoms in there, all of a sudden, you begin to have this new phenomenon in there. So what does this mixing entropy do for the nanomaterial synthesis? And this is where, if you have a two-phase, alpha and beta, the binary, you add a third component. The third component, because of mixing entropy, will be dispersed in alpha and beta regardless whether you like it or not. This is why the mining industry exists. And then by the time the red is chosen well, it's going to help you to compatibilize alpha and beta. People know that in polymer blends. But the interesting part is that by the time you go from 3 to 4 component, you begin to have a formulation flexibility. That means the composition, the type of a composition no longer become a singular. You can have a multiple compositions for each additive you have in each phase. Then you still have exact the same interaction between alpha and beta phase. At some point, as you keep going, eventually alpha and beta is going to become enthalpically miscible. Well, they will still form 2 phase. So in this case, is delta G goes to be a delta S is entropy driven, even though you have all the chemistry. You no longer rely on your repulsive interaction to generate nanostructure, but rather rely on entropy. What that gives you is a diffusivity. The system mobility is going to increase. No repulsive interaction or repulsive interaction being screened. You can go to interdomain interface diffusion. You give the system to find what they wanted to do. And coupled with flexibility, give you the tunability. The system can adapt. I mean, interface, I can adapt. So what we did is we used this and quantify the system mobility at scale as the nanostructure goes with X-ray correlation spectroscopy. So we basically say as the nanostructure come in, what are diffusion mode and what are the system mobility? We decided instead of going from small to large, nanostructure first, microstructure later, we do a reversible. We look at the microstructure first and then regulate the short range diffusion, locally organization to generate nanostructure. What that gives you is something pretty fantastic. Here are 30 micron films, contains over 200 sheets in there. And what I'm most interested in is the number of defects. The periodicity is large to be suitable for the barrier materials, but this is where you are in the defect. Not only reduce defect density, but also you can control the defect type. And this is really a balance between long range and short range diffusion for this entropic driven system. And of course, the property looks pretty good. I'm just run through this. You can do this in porous Teflon membrane for VOC barrier. You can do this in the PET film for water. You can do this in organic photovoltaic for oxygen. And actually, this is interesting. You can even start to use it to modulate the electrons to prevent dielectric breakdown, so that give you a really high dielectric breakdown strength for energy storage density. All right, so I hope I give you some ideas of what we are thinking. And I think if we are willing, the things is going to go really fast. Nowadays, the capability, the research is not only like little things crawling around. You actually can have an escalator really expand. But the question is, with the capability, what are the design philosophy for us to be in sync with everything we are around? And I believe that there's a spot for everyone as long as we follow the rules. And I have to tell you, I do practice to learn the rules at a small scale. I grow lots of orchids who are my flowers. And occasionally I do go to oceans to look at the marine life. So I have to thank all my people. Nothing could be happening with all these fantastic students. This is the RHP team. This is Nanocomposite. Of course, my wonderful collaborator, IIN is a data science in statistics department. Carlos Bustamante really told us biophysics associated with single chain. I didn't get a chance to share with you. And it just be a wonderful collaboration with Alexander Cass in both projects. And of course, I have lots of collaborators here. And I mentioned Monica, who actually did a visiting professor with me 2016. And some of the work actually was spin off by one of former student Aaron Hall into entropic materials. And with that, I'd like to thank you for your attention. Happy to answer questions.
Time for question. We have people going around with microphones.
That was a really great talk. I have a question regarding your project of protein preservation, where somebody tried with FBS. Have you looked into preserving serum samples from humans, mice?
Nope. we see ourselves as build a foundation knowledge to support other people. So that's part of, I didn't mention it. We have RHP app, which is open source software, so people can use it for the design. The idea is not for us to do everything. It's really to change the way how people think and provide that insight to support everyone else to develop it. So that's why I was really happy with Genentech. Well, others is like, you should sue them. So what are you talking about? Just be happy. Someone use it. Someone is going to benefit from it. Yeah.
More questions?
Hello, thank you for your talk. I had a question about the very beginning talking about the introducing randomness and hitting that range in between of like total entropic control or like total entropy or like only one sequence. So do you know what is a good range, or like what is the optimal range of randomness to achieve these types of valuable structures?
So let me tell you what I think. I think each species evolved to occupy different sequence space. If you look at fungi that evolved under anaerobic enzyme, for example, they are evolved they occupy one particular sector sequence space. If you look at the organisms that live on the volcano site, Yellowstone Park, for example, very acidic, very hot. And in fact, I got brainwashed by my own research. I believe as we live in different regions, we also respond differently, right? People live in a really cold environment, getting to really warm, they will have a period of adjusting. So I think this is part of the reason why we come out of this, sequence analysis, and based on what you input, how you train the model, what particular evolution tree you're following, and you can design your system accordingly. And this is where biodiversity must be preserved, because it's stored information in a different space. And nature has done the most sophisticated experiment for us already. All we have to do is just treasure it.
More questions?
Hi, that was really interesting. I'm just kind of curious about, so when we think about like biological proteins, and there's the general structure elements, and maybe binding, stuff like that, but for some of the key biological activity like catalysis, like certain key residues in very specific places end up being really important and functioning really go away with knockouts. So I was kind of curious in this regime of like semi-random synthetic polymers, how to replicate that kind of effectiveness in the biological sphere compared in synthetic polymers.
Thank you for asking the question. By the way, I didn't ask him to ask that question. The paper is currently under review in Nature, and I hope it's going to have a smooth ride. And if so, it will show up in a few months. Yeah. I think we have a solution for that. And actually, it's all embedded in there already. Yeah. But I won't be surprised if we have a rough ride. Then, you have to just, yeah.
Any more questions? I guess I'll ask one.
Yes.
How biodegradable or degradable is your really random multi-layer?
I had a lot of conversation with this, and actually, as a polymer scientist, I actually went back and looked at the energy cost to synthesize polymers.
Yeah.
First of all, for the biodegradable, I think we provide a solution for the society that you are able to program the latency, the pathway, the speed, and the byproduct for biodegradable with embedded enzyme. I think that's already viable solution. And is that Carbon Bios just had a paper early this year in Nature that they use the enzyme and to put in the PLA, et cetera. So I think there's a good solution for that. But if you're really looking at a lot of other polymers, in particular those carbon-carbon bond-based backbone, I don't think they should be degraded because there's so much energy getting into that. You all know in order to reduce entropy during polymer synthesis, you have to build very strong bonds. And there's exothermal process. There's a lot of energy goes in there. they are useful materials. You know, the connectivity give you the mechanical property, give you a lot of unique property you wouldn't get in the other class materials. Historically, when polypropylene was first made, they couldn't get a really high molecular weight with very good stir selectivity. That's why it ended up in the hula tube, right? Instead of a container stuff. So I would say don't toss the baby together with bathtub and use what you need. And not every polymer should be designed to be degradable. So.
All right. With that, let's thank our speaker again. Thank you so much. There's now a lunch break. We'll be reconvening at 1:20. I also ask that all the speakers and moderators and people who have been introducing speakers come to the stage for a picture. Thank you.